We are Adam Pocock, Brooke Hudson, and William Brown, and we are the Trace Amigos. We are going to be presenting on the TEM Lab. The first objective of this lab was to learn the basic principles of TEM operation. The second objective was to learn the different capabilities of the Philips CM200 FEG microscope. The final objective was to examine two specimens under the TEM, one that was annealed at 800 degrees Celsius for 1 hour, and one that was 60% cold worked. Identify which image was which, and then identify pertinent features on each sample. The basic principle that TEM operates on is that electrons have shorter wavelength than of light. This allows for TEM to provide a significantly larger magnification than that of optical microscopes. The electrons in the TEM are generated by passing a current through a tungsten filament. An anode is located where the electrons are generated, which accelerates the electrons through the vacuum where they are focused using magnetic lenses. The electrons travel through the specimen and create a pattern that is visible on the viewing screen, both a diffraction pattern and real space image. TEM is able to achieve a 2.3 angstrom resolution if the environment is controlled. Foam padding lines the walls in the TEM room in order to minimize acoustic noise. Water is also used to cool the magnetic lenses in the TEM and keep them at a constant temperature. The TEM is located in the basement, and the room has extremely thick concrete floors in order to effectively eliminate all outside vibrations. The TEM is also located as far away from the power supply as possible in order to reduce the effects caused by electric and magnetic fields generated by the power supply. The Philips CM200 is a very versatile tool. It is capable of capturing both bright field and dark field images. Bright field images are useful for learning how to use the microscope, but dark field imaging is much more conducive to finding defects. The Philips CM200 is also equipped with an EDX, energy dispersive X-ray spectroscopy detector, which is useful for determining the chemical composition of a sample. It is also equipped with EELS, electron energy loss spectroscopy, which is used to determine band gap, inner band states, and nanoscale elemental mapping. Lastly, the Philips CM200 is also capable of holography, which can be utilized to map magnetic fields at an atomic scale. In order to effectively capture an image using the TEM, the sample must be within millimeters in diameter. To suspend the sample in vacuum, 3 mm copper grids with lacy carbon are used, as shown in Figure 4. The sample holder, shown in Figure 5, then clamps onto the copper grid to keep it in place. If the sample is not made of powder, a focused ion beam must be used to cut the sample and achieve the appropriate size of the cross-section. To capture the image of the first cartridge brass sample, dark field imaging was utilized, which creates an image without the use of the 00 beam. To reiterate, dark field imaging was used because it was much more useful than bright field imaging when searching for defects. Due to the high concentration of defects, the dark spots on the image, we were inclined to believe that the first sample was the cold work sample, as cold working results in a larger amount of defects than annealed samples. When comparing the image of the second sample to the image of the first sample, it is apparent that the second sample has a significantly fewer number of defects. This leads us to believe that the second sample was annealed at 800 degrees Celsius for 1 hour. An interesting component in the second image that was not visible in the first image is the horseshoe-like dark spot. This dark spot is likely a result of contaminants that were deposited while using the E-beam. These are disadvantageous as they distort the image. The likelihood of contaminants like this being present can be reduced by baking the inside of the microscope.